So hi, yes, I'm Sam. I'm a new Chancellor's Fellow at the Roslin Institute, and I want to talk to you today about influenza evolution and phylodynamics. So first of all, some history of flu from an evolutionary point of view. So influenza viruses are RNA viruses. They are segmented viruses, and they code for around 10 or so proteins. They have two surface proteins, hemagglutinin and neuraminidase, and these define the subtype of the virus. Because the virus is segmented, it's possible that you can have reassortment between the segments. So you can have many different types of subtype, but also the internal genes can swap around as well. Flu viruses have a really fast mutation rate. It's of the order of between about 10 and 70 nucleotides per year. And this is one of the fastest mutation rates. Foot and mouth disease virus also has a similar fast mutation rate. And as you'll see later, that actually allows us to do some quite interesting kind of modelling with virus sequences. Avian influenza viruses are thought to have a reservoir in avian hosts, particularly wild anseriforms, as in wild ducks. From this avian reservoir, there are introductions into domestic chickens and domestic ducks, causing epidemics. But also, as I'm sure you know, there can also be subsequent zoonotic transmissions to humans with pandemic potential. To have a view of flu from a phylogenetic point of view, this is there are essentially three main lineages. There's the avian lineage, which I'll see if I can point on the thing. Excuse me a minute. Oh, no. Back again. 1 minute. I'm just trying to find my laser pointer. So the avian blue lineage, the pink classical swine lineage, and the yellow human seasonal lineage. In fact, what you see here is you see two trees, a tree from hemagglutinin and a tree from neuraminidase. And you'll notice that although the clades are represented in both trees of both genes, there are in fact some differences. So you have the main lineages, but you see here, this is the 2009 pandemic lineage, and you can see that in hemagglutinin it came from classical swine, the classical swine lineage, but in neuraminidase it in fact came from another swine virus, which came directly from the avian reservoir. You will also see that there are human to swine transmissions here, forming these other swine lineages. Okay, so what is phylodynamics? Well, in phylodynamics, the idea is that you can use the virus sequences to try and work out who infected who. This could be literally which individual infected which other individual, or it could mean more generally which location transmitted to which other location, location being a country or a city, or which species transmitted to which other species. And you can do this because the sequences accumulate mutations over time. So on the left we have here some sequences. Each different isolate is a row, and they are aligned so the same things occur in the columns, and you can see that there are mutations. If you make a tree of these sequences, so in the middle here you have the tree of human influenza, and you can see a lovely ladder-like structure. It's coloured according to the year of the isolation, and you can see that there's a gradual accumulation of mutations forming this ladder-like structure of the tree. If you plot the number of mutations, the genetic distance of the sequence to the most ancestral sequence through time, you can see this gradual accumulation of mutations. And in fact, what you're seeing there is a strong molecular clock signal. And it's this strong molecular clock signal that can allow us to do much more detailed modelling. So, in particular, if we think of a timescale phylogenetic tree, and here we have our first sequence and second sequence, and they're isolated some distance apart, and we have an idea of what the clock rate is, we can work out when these two sequences, what the most recent common ancestor of those two sequences actually were. Furthermore, we can add locations to the end of the tree. So the first sequence was taken in location 1 and the second in location 2. And then we can ask, well, which location is the source? Because we have an idea, we know already that the time to the most recent common ancestor is shorter to this sequence than it was to this sequence. And we could have a model to tell us, well, how quick might you go from one location to the other, and therefore, how could you make an estimate of which location this ancestral sequence was actually in? You can actually use the same mechanism to do not only locations, but you can do other traits. So you can do species, and you can also do subtype in this same way. Excuse me. So the idea is that you can estimate transmission patterns from the sequence data by inferring the transition rates along the branches. Now, what I mean by this is, so this is my tree, and at the ends of the tree, I've got the different locations, say, and there are 6 locations here, each one with a different colour. And the idea is I can estimate my transition matrix from A to B here. And when I estimate the transmission matrix, that allows me to estimate the locations of all the most recent common ancestors in the tree, so the whole tree becomes coloured. And also, from inspection of the transition rate matrix, you can see what type of transmission pattern you had. So in this case, you have a star transmission pattern, because here you have high rates of A to B, A to C, A to D, and low rates everywhere else. That's here. So the software we can use to do this is called BEAST, and it's a Bayesian phylogenetic program. So in fact, all of the results I'll be showing you later on are all Bayesian results. I won't necessarily have all the confidence intervals on there, but each result has an uncertainty attached to it. So, given the system of being able to estimate transmission patterns, could you, for example, detect transmission patterns due to animal movements? Well, so to test this, what we can do is we can do some simulations. And I did a simulation of around Scotland. I simulated farms, individual farms, within 33 counties of Scotland. And I said the farms can become, farms are susceptible, infected or recovered, and the farms transmit infection to each other, proportional in fact to the movements, proportional to the real movements between the farms, which were averaged out of the cattle tracing system data. So I was trying to make the simulation a bit realistic, and I was trying to ask, well, could I in fact detect the transmission pattern due to the movements? So I generated a simulation, and I looked to see who infected who, and then from that I generated the trees, and then back from the trees I tried to infer the transmission pattern. However, you will notice that I made the problem really hard. So it's 33 counties, so that's a 33 by 33 matrix. But because I was trying to be realistic, I subsampled my data quite a lot, and in the end I thought, well, that's way too many rates and way too few sequences. However, you can do something. You can use a generalized linear model to help you estimate your transition rate matrix. And what you're doing here is you can say that you believe that your transition rate matrix is actually the sum of some predictor matrices. So, in fact, what you can do is add your predictor rate matrices together, and you can allow you can allow them to have coefficients, and your goal then is to estimate the coefficients of your rate matrix. So in this case, as an example, to show whether it's possible or not, I chose the original movement matrix, which is asymmetric, so also the reversed movement matrix, and also a gravity model, which is it's a model encompassing kind of spatial spread, including the source size and the destination size and the spatial distance between them. And yes, in fact, you can show that you can see, even with all those different rates, you can see that if you generate your data from your movement matrix, you can in fact determine that that the movement matrix is the most appropriate transmission pattern, and you can distinguish it from a spatially spreading pattern. So, given this, that's actually quite enough theory for now. I thought I'd tell you a bit about avian influenza and what's happening recently. So, with avian flu, this is a tree of the whole of avian flu, and you can see that there's two big clades. There's this blue clade at the top and this kind of purple clade with a few other ones at the bottom. And here you can see that really it splits the world into the two parts. So you have avian flu, which circulates in the Americas, and avian flu, which circulates in the rest of the world. There are places where there's crossovers, but there's only a few. And the reason for this really is that essentially wild birds, I don't think they fly when they're sick. So they're not going to fly across here, certainly not. So you have this population of North American flu and population of essentially Eurasian flu. Also within the tree you'll see this is the Eurasian swine lineage I was talking about before, and you will also see the high-path H5N1 lineage, which is in this avian Eurasian clade. Talking about high-path bird flu, well, it's actually been around for nearly 20 years. There are major outbreaks in wild birds, particularly in 2005, and there's ongoing epidemics in Asia. It has also spread in the past to Europe, Africa, and the Mediterranean. And on the right here, you're seeing a big tree of high-path bird flu, and the colours of the tree are representing different regions of the world, and the kind of greeny-blue ones are Western Asia and Europe. Also, it's spread obviously between domestic birds, but also from wild birds. And here's that same tree, but coloured according to host type. So here the blue colours are the wild birds. In January last year, this was the picture of the outbreaks of bird flu. The blue ones are resolved, and you can see mostly there's some high-path outbreaks over in China and Asia here. However, between September and December last year, it's actually spread, and there's some in Europe, which is kind of quite unusual, and actually there's a few in the USA. And now, between January and April, this is the picture. And you can see that there's actually ongoing outbreaks, obviously in China, but also in America. And this is really quite unusual because bird flu doesn't normally spread between the continents in this way. If we look at the subtypes, we also see it's not H5N1 that's been spreading, it's actually H5N8. So in order to see what is this new strain, we've generated some time-resolved trees from HA sequences, and these are sequences which have been deposited very rapidly after isolation on the GIS-Aid database. And here we include subtype, region, and host as discrete traits to try to see the origin of the sequences. So first of all, This is the tree by subtype. This is actually a small part of the tree, and you will see larger parts of the tree later. So you will see the purple ones are the H5N8 sequences here. And these are the most recent ones here at the end of 2014. Including region, we see the European outbreak here and the American outbreak here, and also some Japanese sequences. And we see that they have come from a pool in Korea, which itself has come from China. So, how is this? Look, we have Korea to Europe, Korea to USA. But if we include also the host type, so here we have the domestic anseriforms, the domestic ducks, domestic galliforms as in chickens and turkeys, or the wild birds. Now the wild birds, mostly these are actually wild anseriforms, however there are a few non-anseriforms in here, which is why it's just called wild. And we can see that it's wild birds. It looks like you have wild bird transmission to Europe and America. In fact, we can see this. We can see this a bit more. Before I show you, these are timescale trees, and this time point here, this is about mid 2014. The confidence intervals on that are a few months. So it looks like it's summer 2014 that something happened. So in here, what we can see is this is a view from the North Pole. Right. So this is the world, and the cyan dots are the locations marked on the world, and the small dots, so these are snapshots of cuts through my trees. Now I said this was Bayesian, so I actually have many trees. So it's a cut through many trees, and each dot is cut through the tree, but I haven't even plotted all the dots of all the trees, so this is a sample of the dots. The colours of the dots, the magenta is the domestic ducks, the orange is the chickens and turkeys, and the blue are the wild ones. So we see here in halfway through 2013 that there are some domestic anseriforms here. In 2014, you can see some wild bird transmission through to the present day. In fact, I can show you, hopefully, I can show you better than this. One moment. So this is a movie of the process. We're going to start off in 2013. Each dot is representing a sample through all of those Bayesian trees. Oh, we're not going to do that. One moment. Here we go. And you can see the transmission between the locations. So this is transmitting in Asia. And now you have transmission through to Europe and America. This is just a discrete trait model. So the locations are in fact defined as just those places here. When I run the model, I'll just see if I can run it again, actually. Oh yeah, see if I run again. When I run the model, the points are taking the great circle distance. They're not actually taking the true migration routes. However, the true migration routes for the birds do in fact go, there's a migration pathway here and a migration pathway here. So this migration pathway is called the Pacific Flyway, and this migration pathway is the East Atlantic Flyway. So we know how the strain got there. What further, though, is it's an H5N8 strain, which is a bit unusual. To see this is a tree of the whole of high-path bird flu again, and I've broken it up to clusters. And so these are the high-path clusters, and this is the cluster we were talking about. And you can see, actually, this is quite unusual because this cluster, on the top there, you have the cluster split by subtype and on the bottom by host. And you can see that that there are many different subtypes within that cluster. It's a highly reassorting cluster, and it's actually quite different to the cluster underneath and more similar to these low path clusters in terms of the in terms of the reassortments. So yeah, you can see that much more clearly. So this is the bit we've been talking about, but look, in this whole cluster, you've had so many reassortments compared to this one next to it, where there has been basically none. In fact, when we look carefully, we can see if we match up where the subtype transitions occurred here to occurred here, we can see that the reassortment itself is occurring in anseriforms, which we would kind of expect, especially given the fact that this strain is not, it's pathogenic in chickens, but it's not very pathogenic in ducks. So you can have this type of reassortment, and indeed you did. So finally, when we look to North America, so this is the strain when it's just arrived to North America. It's arrived out of Asia, it's arrived to North America, and already it is reassorting. It gets to North America, and it reassorts with local low-path strains. It reassorts twice with local low-path strains. So, in summary, I hope I've shown you that phylodynamics can reveal transmission patterns. And then we've been talking about the occurrence of this new reassortant high-path strain. We've been looking at long-range transmission via wild birds and reassortment in anseriforms with local low-path strains. Finally, as acknowledgements, the sequences, particularly from the high-path bird flu study, were deposited in GeoAssayed very quickly, often within a week of isolation, and this is from many different laboratories. And together we're forming a global consortium to look more into this. And also I would like to thank my colleagues at the University of Edinburgh and Glasgow. Thank you.